My name is Kate Quigley, and I'm a product marketing lead at Benchling, focused on Benchling support for work done in early development. And today I am joined by my colleague, Derek Halliday. He is the head of product management for the applications here at Benchling. And I'm also thrilled to be joined by Keith, who is the associate director of data systems and analytics at Rubius. Before we dive in, I want to do a quick note on some housekeeping tips. We're here on a fancy platform called Goldcast. There are a few tabs, if you look over to the right, that we hope you'll engage with throughout. One is the Q&A tab. Although we'll have the Q&A at the end, we are eager for you to submit questions throughout. We're hoping for a really robust discussion. You can look at the other questions those in attendance have submitted and also upvote those that look particularly of interest. So by the time we get to the Q&A section, we should have a pretty big backlog, hopefully, to get through. There's also the chat functionality. Please add comments to engage with other participants. Very highly encouraged. We'll have a few polls. When those are live, please head over to the polls tab to the right. And then last, you'll see that there's a docs tab. We've included a couple of resources you can download. One is, has life sciences R&D moved beyond traditional LIMS? And considerations for using Benchling as a GXP system. Our agenda today is pretty straightforward. First, at the top here, we'll introduce Benchling and how it can be used as a modern LIMS for biology. I'll first share some perspectives on the market need for a better and scientifically aware solution. And Derek will jump in to show you how it works. Next, Keith will share Rubius' story. And we're eager to spend the last portion of our time on what will hopefully be a pretty robust Q&A. Now let's dive into an introduction to Benchling and how it can be configured to support work traditionally tracked in LIMS. We're excited to show some of Benchling's R&D Cloud capabilities that provide a modern, connected, and adaptable solution that redefines how R&D collaborates, manages samples, and drives efficient and compliant production. First, a quick introduction to Benchling overall for those who are new to our cloud-native software. At Benchling, our mission is to unlock the power of biotechnology. We support our customers in getting their science and products to milestones and market faster. We're fortunate to work with 1,000 of the world's most innovative biotech companies, from small startups to global leaders across biopharma, agritech, industrial materials, and much more. As a result, we get to see how R&D is changing and how the most innovative R&D organizations seek to use technology to unlock and accelerate that innovation. And what we often see is that many scientists are still typically working in a fragmented ecosystem of point solutions like email, spreadsheets, antiquated or homegrown software, on-prem systems, and scientifically oblivious tools like Asana, Jira, or Google Docs. In particular, we hear about the pains associated with underperforming ELN and LIMS solutions. Traditionally, ELNs have been built to support exploratory, hypothesis-driven experimentations, but they usually do not have process management functionality, aren't sample-centric, and are too flexible to capture the structured data required in development. On the other side, LIMS that are created to drive protocol-driven processes in development are typically too rigid to capture and manage the unstructured data found upstream. Most in our industry now agree that existing LIMS solutions suffer from some major shortcomings. First, they're primarily built for small molecules, which have much more straightforward processes. And these solutions are not able to handle the complexities involved with cell and gene therapies, antibody-drug conjugates, RNA therapeutics, and other advanced biomolecules. They're usually deployed into specific sites or for specific teams, and they do not facilitate collaboration or generate a common data set. And they are by and large too brittle. As processes evolve over time, teams who want to adjust these in LIMS find it overwhelmingly slow and difficult to do with this rigid technology. That historically ELN and LIMS have been disparate solutions, disconnected from one another, causes additional challenges. Some common issues include knowledge transfer challenges amongst R&D teams that's just overly time-consuming, which further strains momentum. Downstream teams lack context from prior work done upstream, and we often hear about wasted time, sometimes 1.5 years or 2 years, rerunning experiments that they know have already happened upstream, but they missed that context. There's a need to recreate a sample lineage from multiple systems, and when it comes to something like filing an IND, for example, it's a really tedious and overly time-consuming process. There are missed opportunities to streamline these processes within and across teams in the meantime. And lastly, we hear from IT leaders that they spend an inordinate amount of time maintaining these legacy systems, creating workaround and patches for changing needs and disconnected data. The most innovative teams tell us that they not only want to work together to prevent or reduce these difficulties, increasingly, their success depends on sharing data effectively throughout the R&D lifecycle. Let's jump into a poll, see if any of these are resonating. What are the biggest challenges as teams move into production for your organization? Take a peek over at that poll tab and see if one or many of these might be relevant to your experiences. I'm really happy to see that so many engagement really does track to what we'll end up hearing from Keith's story at the end. He'll talk a lot about the consistent tracking. And of course, all of the above is something we hear often from a lot of customers. There's not just one kind of challenge everybody's experiencing, but many. We'll keep going back to the presentation. So in today's modern organizations, the assumption is that R&D will work in a shared cloud-based solution that combines the best capabilities of both ELN and LIMS. As life sciences products transition from discovery to development, the nature of the work changes. Companies that anticipate this change and support it with the ability to transfer work, data, and processes across these phases will be able to accelerate their overall pace toward milestone attainment. They'll be able to collaborate and transfer data across teams easily and efficiently, which sounds very top of mind for everybody in that poll. And scientific decision-making is done with a holistic, real-time data set. For this reason, Benchling's R&D Cloud is redefining what an ELN and LIMS can be with a unified, powerful solution. As a cloud-native platform, the workspace is configurable to meet the more flexible needs for those conducting exploratory work, and also configurable to structure the process-driven needs for teams working with more defined protocols. Increasingly, Benchling is known as the industry's leader for the connected software driving collaborative processes throughout research and development. That's why we're excited to share with you about Benchling's capabilities in the R&D Cloud, offering a modern LIMS for biology in particular. This modern, connected, and adaptable solution redefines processes traditionally tracked in LIMS. Teams can design, test, and produce complex biomolecules as a unified collaborative effort. Unlike legacy LIMS, the Benchling R&D Cloud offers continual product innovation to keep pace with evolving processes and science. This LIMS solution is truly moving science forward. With Benchling's R&D Cloud, teams can connect people and intelligence to run experiments, manage controlled processes, and track and trace samples in one shared solution. Let's touch quickly on some thematic use cases. Benchling is particularly valuable to teams seeking to optimize handoffs within and across teams, streamline, connect, and track samples holistically, work compliance and data integrity, make it easier to view and share operational and scientific insights, and to manage a more powerful modern tech ecosystem overall. I'm curious to hear how some of these use cases currently fit into your LIMS needs. Let's hop back over to the poll again. We often hear about the need for greater data integrity, just real-time data, harmonized data, shared ontologies, and I think that's particularly relevant as we start moving downstream. Although overwhelmingly, it looks like all of the above option seems to ring most true. So it's really not a single opportunity, but really the ability to modernize the approach altogether. I want to turn it over to Derek. He's going to jump in and show us how all of this works actually inside of the product. First, he can give us a lay of the land of the different applications that are most relevant to a LIMS user. Derek?
Thanks, Kate. Hey, everybody. My name is Derek Halliday. I lead Benchling's application and product management teams. I'm joining you from a very foggy Oakland, California today. I guess the benefit there is that I get some summer usage out of my Benchling hoodie. As Kate was mentioning, in a minute, I'm going to walk you through a demo of Benchling's modern LIMS for biology. What you're going to see is really a solution that's composed of a variety of capabilities within Benchling's R&D Cloud. And when we bring those together, Benchling really delivers a unique solution for process development, sample management, amongst a number of other use cases. But before we dive into the demo itself, I wanted to quickly highlight some of the product areas that you're going to see and call attention to some new features and functionality that you're going to be able to use. And for those of you who already use Benchling, these are really new features that are part of the applications that you likely already have. For those of you who are new to Benchling, these capabilities would be included in the products that you purchase. And so many of the capabilities I'm going to talk about are found in our productivity hub, which are really applications that scientists interact with as a part of their shared workspaces. The first capability is Benchling Workflows, which makes it easier to plan and execute complex processes amongst teams. Within Workflows, we recently released Flowchart functionality so teams can manage processes that span multiple steps and users. This new feature is especially useful for teams using Benchling for study management, process management, execution, biologic material production, as well as sample and inventory distribution. Second, Benchling's notebook, registry, and inventory applications help manage and track samples and inventory. In addition to managing samples throughout their life cycle, these applications increase inventory utilization and allow for really end-to-end sample traceability across shared workflows throughout R&D. And with our recently released inventory tables feature, teams can now access sample information directly in the context with experiments and processes that they're managing in Benchling Notebook. Next, with Benchling Insights, teams can easily optimize operational and scientific performance with reporting and dashboards. And our Productivity Hub applications and capabilities are made even more powerful from the underlying cloud-native platform and extensible ecosystem. One great example of that is Benchling's robust compliance and control features. These features include audit logs that are easier for humans to read, controls when users modify data after the fact, and improving the granularity and clarity of permissions and access controls themselves. And these capabilities are part of what allow GXP compliant teams to control and track their changes at scale. Teams can also work with Benchling's validated cloud offering to leverage validation support for services in controlled and regulated applications. So with that brief introduction, I'm excited to show you Benchling's modern LIMS solution in action, where you're going to see many of those new features and capabilities that I just highlighted here. So let me share my screen. Here we go. So you can see here that I've already logged into Benchling, and just as a brief introduction or reminder, one of the main ways that I can navigate through Benchling is using the icons on the left-hand side of my screen, which correspond to many of those primary applications that I was mentioning earlier. And here, you can see I've already clicked into our Workflows application, which brings me to this dashboard. This dashboard allows me to see an overview of the progress across multiple different workflows and processes within my teams and perhaps my broader organization. This is where I can see and track the statuses of different tasks and handoffs within each of the processes that I may have modeled. Now, as an individual user, I can easily use this dashboard to see maybe any work that I'm personally responsible for. So what I can do is I can select a Save Search view for my pending sample receipt tasks. And here I can see that I have a few tasks that are pending for me. And these are the types of views that are really straightforward to create within Benchling. This is just that same master workflows view that I was showing earlier with a few filters applied on sample receipt tasks, as well as assigned to me and statuses that are not terminal. And so by filtering on these views, I can see this subset of tasks that are assigned to me. And I can also see relevant information on those particular tasks. Not only they're pending, but including links to information about the particular samples that in this case, I need to complete these tasks for. So I can see the sample vial barcode information and the sample ID. Now, one of the great capabilities in Benchling Workflows is that for these types of straightforward and simple tasks, I can directly execute them right from this dashboard. So in order to do that, I can select those tasks, click Execute. And this brings up a modal that allows me to directly execute these tasks in Benchling. I can see here, this view shows me a list of those tasks and a reference again to the sample vial barcode, information about the sample itself, and in this case, these are all plasmid lots. I can also see information sent to me here that, hey, I need to verify the vial barcodes for each of these samples, and I can go in and complete these tasks, and I can give any of my own comments as a part of that execution. So I can say it looks good. And once I've completed the tasks, I can go ahead and click continue. And as I see, as I'm going through these steps, that Benchling is automatically updating the status of these tasks. Now that I've completed them, I can click submit. And this step closes the direct task execution modal that was open, brings me back to my pending tasks view, where I can see that they've been completed and removed from the work that I need to complete. So that's an example of a kind of simple task that I can really easily execute directly within Benchling. But as we know, most of the work that scientists are doing is usually in the context of a larger, more complex process. And our newly introduced flowchart capabilities within Benchling Workflows enables you to model those larger processes as a sequence or a graph of multiple tasks that are changed together. So let's take a look at an example of one of those. Here you can see an example of a workflow for cell line development, and we'll click into one of these to get into more detail. And by clicking into that workflow, you can see that I've entered a view that shows me an overview of the full process. On the left-hand side of my screen, I can see information specifically about the tasks and samples that are involved for this particular process of cell line development. And on the right-hand side of my screen, I can see a visual flowchart that allows me to see and track all the steps, actions, tasks, handoffs that are a part of this larger process. So I can see at each stage of this process, I'm able to track progress on my task at a high level. And I can easily click into any of the particular steps in the process to drill down to a task level view. So for this example, let's take a look at the transfection step to view more information. I can see now all the tasks that are associated with this particular step in the workflow that have been assigned to me or maybe assigned to other individuals that are responsible for this work. And I can see in this case, a couple of those tasks have been completed and a couple are still in progress. Now, within the context of this overall process of cell line development, each of these tasks, plasmid prep, transfection, et cetera, they're a lot more complex than the sample receipt task that I showed you earlier, where I was simply acknowledging receipt of a sample and checking a barcode. So one of the great things about workflows is that for each step in the process, we can define specific protocols for completion or execution by assigning a specific entry template. And since these specific tasks I'm looking at right now are in progress, we can actually see a link to the entries that we defined for transfection. So we'll go ahead and click into that to view it in more detail. And this brings me to the particular entry in Benchling that walks me through all the work that I need to complete as a part of this transfection step of the process. You can see up in the header that this entry was based on a template. If I were to click through here, that would take me to a blank version of what you see in this entry. For those of you who are maybe more familiar with Benchling, this is something that I can specify in the workflow task schema's execution type specifically. And using entries and templates for workflow execution, it also gives you some significant compliance and oversight capabilities that just come baked in, like being able to send your entries for review and approval. So within the context of this active entry itself, we can see at the top that I have a list of all the tasks that are part of this step in the workflow. This gives me easy reference to the context around the tasks and samples that have been maybe handed to me as a part of that process. In this case, I can see a link to the vial of my physical plasmid lots. I can also see specifically the plasmid lots and host cells that I'm working with. And next, I can see here that I have this very helpful lookup table that allows me to gain information and insights into specific samples or containers within Benchling. So let's say that I wanted to pull some more information on the plasmid lots that I'm working with. I can go ahead and place those in my lookup table. And I can see that Benchling performs a lookup and it surfaces for me key information about the specific container that this exists in, the volume of the container. And I can also see a view into the specific plasmid that we're working with, all the way down to the lineage of the domains or genes that are being expressed here. And this is a great example of the power of Benchling's platform. I can see how the processes that I'm designing and executing, they're fully connected from start to finish across all of my teams. So next up in this entry is another feature that I highlighted in the introduction, inventory tables. And these tables, they allow me to easily access and update my inventory containers and volumes directly from the context of executing. So as I go through the work that I'm doing, in this case, I'm working with these plasmid lot files, maybe I can deduct volume from these two containers. And it makes it really easy for me to keep track of the work that I'm doing, ensure that my inventory is kept up to date. This could similarly be used to add or deduct from material samples or reagents that are being used throughout a particular process. And as we continue through the process defined in this entry template, we get into the next steps involved in completing the transfection. And I've included here a mixture prep table as a part of the template. And that corresponds to a mixture prep and recipe that can be explicitly defined within Benchling. Again, this is another example of the power of Benchlink's platform, where we're linking in a set of steps that are explicitly defined as structured data objects. And apart from the traceability benefit, this really further enables teams to approach iterative method development in a really extremely powerful way. So as I complete my work in this entry and finish out recording some specific information, we can see here at the end that we end up with our newly transfected cell lines at the bottom of the template. And by linking this information to outputs of the transfection task of the workflow, this really closes the loop on this particular step of the process that we've defined in the workflow object itself in Benchling. So in this case, we can see that enables me to have those steps complete specifically. And Benchling has automatically propagated those completed tasks to the next stage of the process, in this case, selection. So the next responsible party is really able to go ahead and have those samples or tasks passed on to them and begin the work that they need to do. So you can really start to see the power of the flowchart workflow. Not only are we able to visualize a particular process, but we can also easily track progress against tasks and handoffs within that process and iteratively develop that process in a structured way using components like entry templates, inventory tables, mixtures, and more. And those composable structured components, they're in large part what helps Benchling become a system of record for many of our customers. And you can really also easily interrogate that system of record to take maybe a slightly higher level view of operational or scientific information using Benchling's insight dashboards. So let's click into an example that provides an operational summary of that cell line development workflow that I just walked you through. We can see here some information about the tasks that are completed by month, tasks by status, or by type, all giving me a high level view of the information about my processes. And I can also very easily do things like highlight failed or canceled tasks to identify bottlenecks or pain points as I iteratively develop a process. I'm going to go ahead and wrap up this piece of the demo with a peek into some of the capabilities that our team are actively working on in Benchling Workflows. And the cell line development flowchart example that I walked through was a great one, but also pretty straightforward and linear process. We definitely know that not all processes look that simple. So to ensure that Benchling can really support more complex and iterative ways that our customers approach process development, we'll be releasing a flowchart builder in the fall that lets you define a workflow in a visual drag and drop interface. It also supports much more complex workflows, including branching or parallel processes. And on the right-hand side of this canvas, you can see the inputs into my process modeler, which are actually just a set of tasks that can be defined by your organization or by your individual team. So let's say that in this flowchart example, I wanted to add a step for analytical SEC. I can simply drag that off of the right-hand side onto the canvas, and I can update my links in the process, and I could submit this workflow for completion. So with this capability, we're really taking the power of the workflows that I demonstrated for you earlier, and we're making it even easier for anyone to define and iterate on, from R&D IT to the scientists themselves. And that's ultimately going to support really another level of efficiency and collaboration as your teams approach their work. So that does it for the demo. Before I hand it off to Keith, I did want to mention that with the exception of that process builder that I'm showing you right now, that's going to be available in the fall. Everything you saw in my demo is available right now if you're using Benchling, and you can take advantage of these capabilities through the combination of notebook, registry, inventory, and workflows capabilities. Of course, please continue to add any questions you might have around the demo, this section, or really anything else within the Q&A tab, and we'll make sure to cover those as we end the session. With that, I'm excited to hand it over to Keith to walk us through how Rubius uses Benchling as a LIMS.
Thank you, Derek. So my name is Keith, and I've been with Rubius for about 3.5 years now. So I'm calling in from Boston, and I lead our informatics and analytics group. So I'll start with just a quick introduction to Rubius. So Rubius was founded in 2014, and we have developed a totally new technology using genetically engineered red blood cells as a new class of cellular medicines called red cell therapeutics. The technology works by expressing biotherapeutic proteins within the cell or on the cell surface, which can be used to regulate the immune system or other systems within the body. We currently have 2 oncology products in phase 1 clinical trials, RTX-240 and RTX-224. So Rubius started working with Benchling in 2016. Back then, Rubius was still an early stage biotech focused mostly on discovery work. Benchling was selected as our ELN to use in the discovery and research groups. Those groups utilize primarily the molecular bio app to design plasmids and proteins, the notebook to track experimental observations, and the registry to track the life cycle of plasmids and viral vectors into proteins and transduced cells. Around 2019, we had recently started manufacturing, and we realized we needed a way to capture and trend data from process development and analytical development experiments. So our goal in process development is to compare many different conditions in cell cultures, usually in bioreactors, and determine what are the best conditions for optimal cell growth, and therefore have the best yield at the end of the process. Once the best conditions are figured out, the next step is to scale up the process to clinical and then eventually commercial scale without impacting the yield or quality of the end product. From an experimental design and data collection perspective, since we are comparing so many different conditions within a single experiment, process development experiments have quite a lot of metadata and assay data captured as samples are collected and tested throughout the process. So we needed a way to build a database that could capture all of this data in a way that was conducive to rapid querying and trending. Engineers and scientists needed to be able to compare different culture conditions across multiple experiments without having to collate it all together manually every time they do their analysis. So since we're already using Benchling in the company, in the research and discovery groups, we decided to expand its use into process development as well. So we started out with just the notebook and registry apps. Both of those tools are pretty flexible, so we were able to design a data model and data entry process that closely matched our experimental design. It was able to capture all of our data in a structured way. Since then, over the last 3 years, we have built upon that initial foundation, and now we're using pretty much all of Benchling's applications across the daily functions in process development and analytical development. So next I'll step through some of our use cases for each of the different applications. So we'll start with the notebook and templates. So first of all, we make sure to configure all of the permissions and access policies so that each department only has access to exactly which notebook templates and which folders are relevant to them. This streamlines the user experience a bit so that people don't get overwhelmed by seeing all kinds of stuff that is only relevant to other departments. Templates in general help to standardize collection of metadata and qualitative observations across the various subgroups in each department. And then we also use the mixtures templates to define recipes for media and buffer preparation. And then when the end users actually go to prepare those solutions, those templates actually walk them through the preparation in the lab. And then finally, we use the notebook sign-off and peer review functionality to capture proof that we are actually reviewing our data. So next is the registry. The registry is Benchling's customizable database. So we use this to standardize and build relationships within experiment and sample metadata and capture assay results for all of the departments. One example would be raw material genealogy. And what I mean by that is, for example, which donor cells and which lots of reagents went into each cell culture in each bioreactor. And then the registry automatically pushes all of this data that you enter into Benchling's backend database so that it's easily searchable and queryable by managers or data analysts or whoever need to see it. So the inventory we use mainly for keeping track of our raw materials. Some examples are inventory location tracking, for example, which freezer and which shelf in which freezer is each raw material container in. We also use Benchling's labeling functionality to print labels onto all of our containers that have all the relevant metadata, as well as barcodes. We use the inventory for stock tracking, such as which reagents are running low and need to be replenished, and also which items are nearing their expiration dates and should be discarded. So the Workflows app, we use that mostly for managing requests submitted between different departments. So for example, our process development group will submit samples to our analytical development group for testing. We use the Workflows app to indicate which samples need to be tested, where they are located in the lab, and which tests need to be performed on those samples. Another example are solution preparation requests. So we have a group in our company that pretty much all their main job is to just prepare solutions requested by the other departments in the company so that they can perform their experiments. So they're making things like media and buffer, and then they hand that off to the other departments. So we have the workflows set up to indicate which group is requesting the solution, which type of solution is needed, when it is needed by, etc. Also, it works well for managers because the workflows, it has a lot of like timestamps and status changes involved in it. So it's pretty easy to track metrics around the requests, such as what are the average turnaround times and how close are we to capacity and staff workload and things like that. So Insights is Benchling's built-in visualization tool, and we use that to rapidly visualize data from pretty much all of Benchling's internal applications. An example would be comparing small-scale process development experiments versus manufacturing scale runs. Another example is calculating process averages across multiple different, maybe hundreds of different experiments in the notebook. So you can maybe get an average of an entire process within a program. We also use it to graph usage and productivity metrics, such as how many notebook entries each group creates per month, how many samples each group is testing, how quickly notebook entries are being signed off and reviewed, and average turnaround time for requests. So the warehouse we use the warehouse together with Spotfire. So if you're not familiar with Spotfire, it's a visualization and analytics platform. So we use the warehouse together with Spotfire to aggregate Benchling data with data from other platforms, and then analyze all that data in one place. So what are the lessons and best practices that we've learned over the years? So when it comes to collaboration between departments, in general, process development and analytical development usually require data to be much more predefined in its structure than research and discovery. So what I've found is basically the earlier you can define your metadata structure and assay result structure within templates, the better. I know that's easier said than done, because a lot of times in early stage development, it just, you don't even know what you're measuring yet. But the earlier you can figure that out, the easier time you're going to have. Also when you're designing your data model in Benchling's registry, be sure to configure entity links in your schemas to build relationships between data that's coming from different departments. Then when it comes to handoff between departments, make sure to use workflows. It just makes it much more automated, and having automatic notifications to each of the stakeholders in the relevant groups, and having all the data or all the appropriate information that they need to perform that task right in front of them. So next, for operational considerations, you want to make sure to configure your access policies and permissions so that they're specific to each department or even each subgroup in each department. This provides a more intuitive user experience for each team, and prevents users from being overwhelmed by seeing a whole ton of stuff that doesn't matter to them. It also makes, it can also be a compliance consideration, just because maybe you don't want certain departments to see certain categories of data. And then regarding aligning stakeholders and gathering requirements when you're building all this stuff out, I recommend finding a power user or champion in each department, and then try to funnel all of the all of the work through them, all of the requests through that person, so that they can sort of triage it and determine what is most important, and then use that person as your main point of contact. And then lastly, you want to make sure that Benchling is directly accessible at the bench from within the lab. You don't want users to have to jot down notes on on sticky notes and then bring all of that back to their desk and enter the data. So it's better to just have either laptops or some sort of way to enter the data directly in the lab. So for the technology ecosystem, you want to make sure to select tools that allow for integrations and communications with each other. You don't want any of your data to be siloed in a single platform. Next, you want to try to use scientifically aware tools when possible. So for example, I often see people use SharePoint, not only to store data, but to manage requests and things like that. SharePoint is a great generic tool, but there are much better tools available for these use cases that are actually designed to be used by scientists. So what's next? So now that we have built, so for our pipeline, now that we have built this framework in Benchling, as new potential products come through our pipeline, we can continue to use the same framework we have already fine-tuned in order to hit the ground running when it comes time for process development. So it may take a little bit more time, for your first few products that you run through the system, because you're still fine-tuning it, but you can reuse all of that work that you've done, all that investment, and it'll just make your next program that goes to the pipeline will be much faster. So another thing that currently we are in the process of migrating into the new workflows application that Derek demoed earlier. We're very excited about that. The main reason is because compared to the legacy workflows application, the new version of that application allows for much more granular control and tracking of individual subtasks within a request. We're also looking to convert some of our notebook templates into those new workflows, just to make the process of entering the data and going through the experiment even more intuitive. So, and then lastly, we always plan to try and improve our efficiency. So we plan to utilize additional trackable metrics enabled by the workflows application to further enhance our operational efficiency. And lastly, we want to try to build out more direct integrations between the instruments in the lab and Benchling. Using Benchling's lab automation platform, we still haven't really used it that much. So yeah, we really want to start using that. Cool. So that's about it. Thank you all. And now I will hand it back over to Kate for the Q&A.
Thank you all for submitting questions throughout. I think we've got a good number already here. So I'm going to go ahead and start, I think, with the first question at the top here. This is, I think, primarily from you, Keith. Do
Sure.
Rubius's research, process development, and manufacturing groups work in a shared version of Benchling? And if so, can you share a little bit about how that works? I know some of it's proprietary, but kind of whatever you are able and comfortable to.
Sure. Yeah. So I guess the short answer is yes. We only have one Benchling environment, so every department that's using Benchling is in the same environment. And so in order to make that work, really, you just have to be very good about setting up your access policies and making sure that each department and each subgroup has their own team. In Benchling, it's called a team, and then you can apply permissions specifically to that team, to each different team. And you can make it very specific, down to the point that when each of these groups log in, they'll see something completely different. So, for example, like maybe you want the research group to be able to edit metadata on a sample after it's been submitted, but you don't want the analytical development group to be able to do that. So, by leveraging teams, you can set those permissions very specifically. You can also even hide entire apps. Like maybe one of the groups doesn't need the inventory or something like that. You can just completely disable that app for them. So it works pretty well, as long as you go through that effort to set up those permissions.
Thanks, Keith. Next one on here we have is Benchling GCLP compliant. I'll take a quick pass, and maybe if Derek wants to add anything else. We have customers currently running de-identified biosample information through Benchling today. Over the upcoming year, too, we have significant investments to build out a lot of our GCLP compliant capabilities. So we can go through that in more detail in account conversations for those who are seeking to use Benchling increasingly in that space. Derek, is there anything else you want to add in there?
I think you covered it pretty well.
Okay. Great. Actually, I'm going to even jump down because I think there was a somewhat related question in here. Are there features in Benchling made specifically for the quality teams? And if so, what are they? I think you showed a little bit, and we can also talk about where we're also going there.
Yeah, I can definitely cover this one. I mentioned a couple of them at the top of my presentation, where we've been making some pretty significant improvements to our audit logs, audit trails, as well as improving access controls and role definitions there. And we also rolled out recently a new feature called data modifications, which really prompts users for a reason for change when they're changing data that's already been entered. So all this is really helping us as a platform be more ready for those types of use cases Kate just mentioned, and provide the capabilities that quality teams need for oversight. This is actually a very timely question from my standpoint, because right now we're in the process of defining how we continue to push that forward and build both exports as well as even in application capabilities for quality teams specifically. So I know this one was submitted anonymously, but if the LIMS poster is interested in talking more about this, I'd encourage them to reach out. And it's, again, an active area of development for us.
Thanks, Derek. Next, we have a question for Keith. And there's a number of questions, I think, probably for Derek, also around kind of integration. So we'll share this one around the ability to integrate with, I think, FreezerWorks in particular. I think that's pretty common use case, or other inventory systems.
Yep. So good question, Mark. One of the benefits of Benchling's inventory that I kind of showed a little bit in that demo is that it is intimately integrated, connected to the other parts of Benchling. So the first part of this question, in terms of inventory adding, removing links to other parts of Benchling, absolutely. That's a big part of the benefit of using Benchling's inventory. In terms of third-party inventory systems, one of the other strengths of Benchling is our extensible platform, our developer platform in particular. And so this is an area where if it's not out of the box, we almost always have a capability to integrate with a third-party system that may be a part of your chosen ecosystem.
And while we're on that, I think similar, are we able to assign a barcode to storage units? I think a lot of scanning and barcoding questions are always very relevant. Thanks, Sadiq.
Yep, there were a few on this one. I think I answered one of them asynchronously too, around a recently announced integration with Bartender for label templating as well. But this type of integration is pretty essential and key to Benchling's use as an inventory. So I'd say the answer to this one is absolutely. There's native integration with those types of systems.
The next question is from Lindsay, and I'm actually going to ask Keith if you can share a little bit around how you use Benchling for data analysis, and then also probably you're integrated with other tools. You talked a little bit about Spotfire, but can you do a little bit of a double click into your experience there?
Sure. Yeah. So I guess the two questions on here are yes to both of them. So Benchling does have built-in data analysis functionality. That's the Insights app. I think, not sure if Derek showed it, but yeah, basically it allows you to plot any data that's in Benchling in, like, a line plot, a scatter plot, a bar graph, whatever you may need. That said, that only works for data that's internal to Benchling. So if you have a use case where maybe you have a few other systems where you need to pull data in from multiple different systems, Benchling's built-in tool won't work for that. So you'll need to use the warehouse, which is basically, it's a SQL database, a PostgreSQL database that pretty much almost all of the data that goes into Benchling gets pushed to that database, and that database is very easily queryable from any other tool that can query PostgreSQL. So in our case, we use Spotfire just to query that warehouse, and then Spotfire has a very advanced statistics and other types of data aggregation, data wrangling functionality built into it, and it can also merge data between other sources. So we actually use both for more visualization and analysis that is just specific to only Benchling. We use Benchling Insights. If it needs to pull other data in, then we use Spotfire with Benchling's warehouse.
I might add one more comment on that one in terms of like an area of more that's coming, which is the types of use cases that Keith just mentioned in terms of putting a third-party tool on top of Benchling's warehouse, super common practice by our customers. We're also in progress developing capabilities to have more direct integrations with third-party point solutions, maybe statistical analyses tools that you might be familiar with. So one that we're actively in development on, and I just recently saw a great a demo internally as a two-way integration with Jump for statistical analyses. So supporting more natively both pushing and pulling data sets and analyses to and from Jump. It's going to be something that is coming soon, probably early next year.
Great. I think we probably have time for maybe one or maybe two. Let's see how we can do. This question is from Andrew around design of experiment or multivariate analysis. I think we've talked a little bit about analysis. We could start in there, and then we can talk a little bit about DOE. Derek, do you want to jump in here? And then Keith, if you also have some experience here, love your anecdotes too.
Yeah, this is an area that we're also actively working towards. I think that one of the things that we're excited about is the ways that more complex workflows are enabling us to sort of model processes that are actually reflective of what our customers are actively doing. In terms of DOE specifically, again, we're approaching this from a standpoint of some robust capabilities in Benchling, but also integrating with third-party statistical design tools like Jump that may be useful for this part of the process. One last thing I might mention is that for some specific multivariate analyses themselves, we're also exploring applications of machine learning on data sets within Benchling to help our scientists actually perform some of this work directly within the application itself. Keith, would you add anything from your standpoint?
I was just to say, I see we have 30 seconds from the end. So I just want to go into next steps really quickly because I think there's some good opportunities. For those who want to continue the conversation, we are always encouraging reaching out to schedule a one-on-one demo. If you're a current customer, please reach out to your account team. And if you're not, you can email me directly and I can connect you with somebody. My email address is kate.quigley, Quigley, @benchling.com. And I want to thank everybody for attending today. We'll send a recording and please pass it along to your colleagues. Thanks so much.
Thank you, guys.
Thank you.